today, but it's kind of an extraordinary story. I think the public really appreciates the cost and the time to sequence the human genome over the last decade. $22 million to $2,000 per genome. We're able to now record from hundreds of cells at a time. A decade ago, which was doing at most, I think, 16 or sometimes 96 cells in a very special way in vivo. Smartphones now have 190 million and counting. Technically, really a revolution in what technology might be able to deliver in terms of the conveyance of information and computing power. Following Moore's law has gone up eightfold in that time. Now, the version of Moore's law we like to show is the blue line here. Let's give you a sense of how things have changed for the microprocessor industry here in Silicon Valley, and it has, in fact, since 1965 pretty much followed the predictions of Gordon Moore that we had a doubling of processing power every 18 to 24 months. But you look at that number, the reduction in cost for genomic sequencing that I said instead of 20 million instead of 2,000. You can get a sense of that over this very rapid period in the mid-2000s that things began to plummet. And the result was that suddenly we were able to identify genetic associations with many of the psychiatric disorders. It really took off just about the time that genomic sequencing became cheap. And it took off partly because we had put together large repositories of DNA in patients who were very carefully described. We're now up to nearly 1 million samples across 9 disorders, and this is a wonderful international effort involving 32 countries, sharing data, sharing ideas, and sharing results as well, to be able to give us a sense of where the findings are. When you look at those findings, it's really quite surprising that the candidates that all of us had expected for schizophrenia or autism or depression were the ones that were tied to medicines that we had been using. So when people were looking at genes associated with serotonin or dopamine, there have now been hundreds of findings, as I showed you in that previous slide. And it seems to have just about every system you can imagine except for serotonin and dopamine, but still seem to show up anywhere in these kind of unbiased approaches to figuring out what might be the underlying risk architecture of these disorders. But the candidates that do come forward are really interesting. They're largely developmental genes, the genes that are expressed in the brain, specifically usually in the second trimester, sometimes in a big way in the third trimester. They may be genes that aren't expressed in the brain so much in adulthood. But it's very interesting to help us realize when you look at the genomics, you're being pointed towards pathways that are either really important for immune function or pathways important for neurodevelopment. Without taking a lot more time to tell you this, I wanted to just kind of summarize what I think are the kind of key messages that we can now understand about this. The first is that this is a story about risk and not cause, that the genetics of mental illnesses or neurodevelopmental disorders is going to be remarkably complex. It's not what we might have thought a decade ago, where we were looking for a single gene that would explain what causes one person to get schizophrenia and not another. There are so far at least 108, and we're still at the very beginning, genes that have been shown to have variants associated with risk for schizophrenia. The actual number is likely to be hundreds. And it will probably be a little bit like height. This is going to be the result of many, many, many genetic variations working together. But what they do is they bias towards a particular set of maybe cognitive features or particular way that people may perceive the world. And then it has a lot to do with the environment, especially the environment during development. It determines how that bias plays out. So this is about risk. It's not about determination or a cause. It's very clear that the genomics is teaching us about the importance of timing. As I mentioned, most of the variants that have been of greatest interest are ones that may be expressed in the brain, specifically during development. And that tells us that we have to be thinking about how this plays out over time, and having a developmental perspective, even for understanding genomic impact, is going to be critical. And finally, something that that there is tremendous heterogeneity in your body for your DNA sequence. It's not just that each of us has slightly different DNA, unless we have an identical twin. It's that even within each of us, cells can have very different DNA sequence because if cells are replicating, they make lots of mistakes. And they make more mistakes when they replicate quickly, and there's no place where cells replicate more quickly than in the second and early third trimester of development in the human brain. So you can look, as we have done, for DNA variations that are associated with these diseases in blood, but they could be very different in brain. And we have to remember that at the end of the day, even though we have no way of doing this in living tissue, that brain DNA can be quite different than blood DNA. Move on to the next revolution, which I think is in neuroscience. This is the era of the Brain Initiative. Lots of excitement about this, and you heard a little bit about this in the previous talk around mapping white matter and what's called the Connectome. The Human Connectome Project is this very interesting effort that's been underway for about 6 years now to take a large number of subjects, in this case, either 1,500 or 1,200 healthy individuals who volunteered to have very, very extensive imaging of their brains. These are people who have had all of these different modalities to look at structure and function and chemistry, even sometimes molecular imaging, along with lots of other tests of their cognition, their personality, genomics, a whole range of things. And all of this has created this wonderful public project called the Human Connectome Project, with all of the data being made public. In fact, it's been made public as they've gone along. So they released the first 100 and the first 500. They released 900 about 3 months ago, and they'll be releasing the entire data set, which is now complete, in the next few months. It's a fantastic opportunity to look at kind of a reference atlas. In the same way the Human Genome Project gave us that picture. What does the human genome look like, and not only how is it different from a mouse, but how is it different between each of us? What's the kind of normal range of variation? We're doing the same thing here with the Human Connectome Project, getting a sense of both structure and function and how it differs between any two individuals. Unfortunately, this movie isn't showing, but the kinds of things that it's already told us is that there are these very interesting relationships between different areas of the brain. Real estate that we had no way of thinking would be connected that are showing up, particularly with the brain at rest. And so it's given us, in terms of these functional connections, entirely new insights already into how the brain might be organized and how information may be encoded. So it's still early days with this, but a very exciting time. Now, imaging the developing brain is quite a challenge, but people are now undertaking that challenge in a number of different ways, and I wanted to take just a moment to tell you a little bit about that. The figures on the right, the images are from the top row, neonates, the second row, the 1-year-olds, and the third row are 2-year-olds. And it's looking at what's called the default network, which is just the brain at rest. The curious thing about this is that when you look at brain activity with fMRI, the time when you see the most activity is when the brain is, quote, at rest. Unlike any other organ, that's when there's the greatest metabolic activity and the greatest general sense of involved real estate, let's say. And then it goes away when you challenge and give a task, because areas take off and the brain begins to focus in. But you can begin to see that process develop, and that's what the studies are on the right-hand side of the slide. The left is a wonderful image from Rebecca Sachs at MIT, who's been studying this very development, but now in a social context, and how mother-infant interactions can alter the pathways for brain development. The story I wanted to tell you is one that's actually just come out this week, and I think it's a really extraordinary story about how we can now begin to study brain development in young children. It's from Patricia Kuhl's lab at the University of Washington in Seattle. And what the TAC's been doing for many, many years is trying to understand language acquisition and the difference between monolingual and bilingual families. And what she's done here is to take 11-month-olds who are not yet speaking and put them into this MEG scanner, magnetic encephalography scanner. You can see that wonderful picture in the upper right hand there of an 11-month-old very happily in this non-invasive, absolutely quiet instrument, which looks like it's something from the 22nd century, perhaps. And what they've done then is to play syllables while the baby is in there. NMR, which is what you see in the legend for this figure, it means the mismatch responsivity. So they're playing either English or Spanish syllables, and then when they switch from English to Spanish, they're looking at the brain's response. And what they're showing in a very interesting way is that even at 11 months, kids who have grown up in a bilingual family and have been exposed to both English and Spanish are processing English and Spanish differently, but I find it remarkable that they can already do that. But they're also not only identifying the difference in the language, they're identifying both English and Spanish syllables as linguistic, and they're beginning to separate them out and to respond accordingly in a way that doesn't happen for a monolingual family. So what Pat and her team is interested in doing is using brain imaging, in this case with MEG, magnetic encephalography, to begin to look at this process by which language is acquired in the brain, not only behaviorally, but now in terms of neural pathways. It's really an extraordinary opportunity because kids at this point can't tell you what's going on in their heads, but you can begin to get a little bit of glimpse into that black box. So where are we in the case of neuroscience? Well, I would say that there's no question that we have a sense of how the brain develops structurally and functionally. We still have lots of unanswered questions there, but the most remarkable is to realize that this process of brain development goes on well into the 20s. It's one of the reasons why the insurance companies don't rent anybody under 25. They realize that the frontal cortex isn't quite there until age 26. We spent a lot of money at NIMH proving that, but we could have simply asked first and we would have found out the answer much more quickly. Increasingly, we've begun to use these tools to identify that there are circuits that are altered in mental illness. And even though we still call it mental illness or behavioral disorders, these are in fact brain illnesses. The difference from neurological illnesses like ALS or Parkinson's or Alzheimer's is we're not talking here about dead cells. We're talking about circuitry. Sometimes people call these connectopathies. There's a problem in the connections and the functional relationships within the brain. But indeed, the results are no different than if you were to compare having a myocardial infarction where you killed off a bunch of heart tissue to having an arrhythmia. These are essentially the arrhythmias of the brain. And with these kinds of tools, we're able to finally see that. And it's been just remarkable to use the tools we have now to understand plasticity and resilience in a way that none of us could have imagined. The brain is this extraordinary resilient organ. And in fact, what we've come to learn is that most of the time if you have a brain illness with symptoms, you've had problems for years and years and years, and it's the emergence of symptoms to the point where the compensation is breaking down. It's not when the problem itself first develops. And that's an important insight because it tells you that when you see a change with neuroimaging, that might not be the lesion, that might be the opportunity or the attempt of the brain to compensate for that lesion. Let's finish up by saying a little bit about technology, which is, I think, really what Dennis was hoping to hear about. And I'll take you through this in a couple of different ways. I think there's just so much to say here, and it's such an interesting time because it's still really just unfolding for us. It has been phenomenal to realize that in spite of technology revolutionizing entertainment and information and banking and commerce and so many other things in our society, it really hasn't had the impact on healthcare that one might have expected. And why that is, we can talk about. There are many, many reasons, I think. It doesn't say that it's not to say that it won't have that impact. I think we just haven't really yet found that the sweet spot where this is going to work. I'll give you a few kinds of stories that just remind us of what we can do today very quickly. This is an old story, more than a decade old, about Iris Murdoch, a famous author from the UK, who wrote some 20 or so novels in her lifetime. And developed Alzheimer's disease late in her 70s, but continued to write. And her final novel, which is called Jackson's Dilemma, is shown with the triangles there, is the one that she wrote long before she was diagnosed with Alzheimer's, but at a time when she was already showing manifestations of very subtle manifestations. One wouldn't have picked it up clinically. She probably didn't pick it up personally, but you can see it by analyzing the text of this novel. Really extraordinary. What this shows is that if you map the emergence of new words as you go through the book, going from 10 to 20 to 30 to 40,000 words that have been written, and you identify when new words come up. You can see in her earlier novels, Under the Net and The Sea, there was a continued trajectory that went on even as she got into the much later parts of the book. But that begins to break down in the earliest stages of Alzheimer's. She was diagnosed some 4 or 5 years later. But I put this up because it tells us that there are really interesting behavioral manifestations or cognitive manifestations that are out there all the time that we don't often take advantage of. Text analysis is fast now, it's cheap, and it's highly accurate, and allows you to do these kinds of techniques. Is this a diagnostic for early Alzheimer's? Well, not everybody's a novelist, so it probably wouldn't be that useful, but it gives you some sense of the opportunities that could be out there if we think differently. Here's another example of the same sort of thing, and unfortunately, because the animation is off, you can't see this, but search term, I just did this, I think, last weekend. I put in suicide as a cause of death into Google search, and I got this peak in August of 2014, and the quiz question was what happened that week, and as you can see from the answer at the bottom, it was the week that Robin Williams killed himself. There is an enormous amount of information that can be mined out of search. These search trend analyses, any one of us can do it. This is one of the options that are sitting there within Google search. And you can put in your favorite term, your favorite question, and within seconds go through billions of data points to get an answer like this that tells you something about what's happening in society at a given point in time and allows you to follow that. Lots of devices on the market. There's your EKG machine in the upper left, your otoscope in the upper right. On the bottom left is a contact lens that's not yet available, but it's being developed by my company, Verily. It's a contact lens for people with diabetes, and it has a glucose meter embedded in it, a tiny, tiny battery, and a Bluetooth transmitter as well. So if you have diabetes and you wear contacts, this gives you continuous readout of what your glucose is doing, and it sends that to your phone, and your phone in turn can control your insulin pump, and you have already a closed loop like an artificial pancreas. So the kinds of things that can be done here are extraordinary. Google Glass, which Dennis is backstage now, but Dennis has been using as a social prosthetic in kids with autism. Lots of opportunities here to think about how devices could make a difference. Perhaps the most powerful one is the one we all have in our pockets, at least 190 million of us in the United States, and that's the smartphone. What a really interesting opportunity to capture what somebody is doing every day and what they're thinking about, and the opportunity to look at activity and sleep and to look at how people are typing. When you're depressed, you develop psychomotor retardation, or when you have Parkinson's, the problems with the fine motor skills.